Yeah. Is that okay? Good morning. And we have been talking about the characteristics of organisms. Good morning. Characteristics of organisms, right? And characteristics of organisms, we discussed this first part on a day, Sunday, previous week. And we will be continuing that today. We discussed about the respiration, and it is the cellular respiration that we want to discuss. Cellular respiration take place in two ways, maybe aerobic respiration with oxygen or anaerobic respiration without oxygen. So respiration, there are two experiments we have to do under the cellular respiration. The first one is to see whether carbon dioxide is removed. Carbon dioxide is removed as a product of respiration. In cellular respiration, we know that glucose, C6H12O6 with oxygen, carbon dioxide, and water vapor is removed when you balance it with something like this, and energy is produced. This happens in the mitochondria in a cell. So glucose is there and oxygen. So we want to test in this unit that oxygen, to show that oxygen is required and carbon dioxide is produced. That is what we want to test, experimentally prove that that oxygen is required, carbon dioxide is removed. And to show that oxygen is required, there is a little experiment, and the questions can be produced, generated from that part. The next one is that carbon dioxide is required. So the test for oxygen, requirement of the oxygen is tested with the experiment, if you can remember the experiment by using this one. And we'll see how it happens. Normally, when an experiment is done, the experiment is done by two sets up, right? Experimental setup and control setup. So the experiment can be controlled experiment. It can be controlled experiment. The control experiment has two sets up. One is experimental setup, and the other one is control setup. In the experimental setup, right, we do the experiment by using by giving the particular components, also by keeping all the others constant. The particular testing variable factor is tested here. In the control setup, we don't give, we avoid the required part. we avoid the required parts in experimental setup. In the control setup, we give that. For example, I will show you how this happens. If you can see that, if you can remember the requirement of carbon dioxide for photosynthesis, this is grade 11.1. This is grade 11.1. There is a plant and it has leaves. So it has leaves, right? We are going to see that carbon dioxide is required. Carbon dioxide is required for photosynthesis. So we can cover this one and then we put potassium hydroxide solution. Potassium hydroxide solution, and this one we put water. So now you can see this is a controlled experiment. Controlled experiment because carbon dioxide is removed from here by potassium hydroxide solution. Carbon dioxide dissolved in potassium hydroxide solution, and so that is experimental setup. Experimental setup. This is experimental setup because the variable, the required factor of carbon dioxide is controlled. That's not given. Control setup is the one that everything is given. So you can have carbon dioxide, but water is given as the only we want to avoid one factor, not two factors. If you avoid potassium hydroxide and water, both are there. That's why AQ refers potassium hydroxide and water. Both are there in this bag, right? So what we want to avoid is only potassium hydroxide. So potassium hydroxide is not there, but water is there. So if you say 15 milliliters, if you get 15 milliliters, you got the 15 milliliters of water as well. Only potassium hydroxide there, so carbon dioxide is removed from that. So this is control setup and this is experimental setup, right? So as the result is that after keeping this one in the sun, and testing is done for the starch test by using iodine solution. There's a process that we have to go, and we can see that this one does not respond to the starch test, but this responds to the starch test. So then we can see that carbon dioxide required for the photosynthesis process. But it is not what we have to discuss here. This one, it is to see that oxygen is required for the cellular respiration. And it has to be shown by using a controlled experiment. By using a controlled experiment. In the controlled experiments, you have experimental setup and control setup. And see how we can do that. According to your textbook explanation, it is done something like here. It is done something like here. So the setup are arranged by using this instrument, this laboratory equipment, right? So the inside the container, germinating seeds are there. You can have the germinating seeds. Germinating seeds are there, and then there is a small tube, maybe a boiling tube, you can say, and put potassium hydroxide solution there, KOH, aqueous, and then you can see, yeah, you can see water, colored water is available, and this is closed. That is one setup, and the same thing is prepared again, only the thing that there you don't have this one. So the conical flask is used. Is that conical flask? And yep, there's one part over here. So then this is covered. So you can have again the glass tube, rubber tube. It should be rubber tube, and then glass tubes are there with the colored water, right? As usual, the colored water is there. And germinating seeds. As usual, the germinating seeds are there. So this is the experiment test. Experiments is here. And glass tube, germinating seeds, right, rubber. Potassium hydroxide is there. In order to give the exact part, right, so you have to use again a test tube and put some water. If it is to be exactly the balanced one, then here you can have water, H2O, and this is KOH aqueous, right? Then the colored water is available here, right? And colored water is available here. Because of the capillary action, a little amount of movement will be shown. Not exactly the same level of water. With water, there should be a little one that is we call the capillary action, right? Two forces are acting on to move upward. That is cohesive forces and what is the other one? Cohesive forces, adhesive forces. Yeah. Cohesive forces and adhesive forces, it moves a little up. But here, if you consider the total height is 5, if you break this height into 5, 1, 2, 3, 4, 5, it should be equally divided. My division is wrong. So you can have 1, 2, 3, 4. But there are 5 parts are there, and the height that water column goes is 1 upon the 5. 1 upon 5 height is there. Right. Now let's consider what this experiment is and how. The reasons to use those things. And I told you that this is actually a controlled experiment. Controlled experiment. This is control setup. Control setup that everything is given. And this is experimental setup. In the experimental setup, we avoid carbon dioxide, right? Why carbon dioxide is removed? What is the reason for removing carbon dioxide? Well, this is the story. This is the story of this experiment. How many of you are in need of explaining this one? I just want to check. Can you put a little, raise your hand? Yeah, we want a little more explanation. 1, 2, 3, 4, 5, 6, 7. Okay, many. Good. All right. Okay. Thank you for that. And see, these are the reasons, right? 1, why do you use a rubber tube? Why do you use a rubber tube here? What is the use? What is the use of having a rubber tube? What is the use of having a rubber tube? Can anybody explain or give me reason just early in the morning, like 4 o'clock here? What is the use of a rubber tube? No idea?
You can block the entering of the air.
Well,
Easily.
without this rubber tube, you can straightway use this one, right? Without rubber tube, no rubber tube is used. Why? Without rubber tube, you can go with that one.
To maintain the pressure inside the conical flask.
That's correct, exactly. Maintain the pressure. At which occasion? In which occasion?
When fixing the lid.
When fixing this cork.
Cork lid.
Yeah, cork lid. Why? Maintain the water level. It is practically difficult, right? Practically difficult to fix this one because when you tightly fix the rubber cork here, right? Rubber cork, then there is internally air pressure. That because of the air pressure, when you fix this one to water, right, this will not be like this. This will be putting down because of the air that won't raise upward. In order to maintain natural one, so you first open the rubber tube, right? Open the rubber tube and then fix without any problem. You can get the water level a little up because of the capillary action. And this one also will be not like this. At the stage, it will be here. Then you tightly tie up this. Tightly tie up this after fixing it. Now, no air pressure inside. Okay. Then, right, that's all. And after that, what is the use of potassium hydroxide? What is the use of potassium hydroxide? This experiment setup should have this one also. What is the use of potassium hydroxide?
Removal of carbon dioxide.
Yeah, to absorb, to remove carbon dioxide. But our experiment is, you know, that the requirement oxygen for respiration. Requirement of oxygen for respiration, but we remove carbon dioxide. Why? Why? Why is that? You know that the reason is the experiment, the cellular respiration, aerobic respiration takes place like this. C6H12O6, very well taken by the seed. The seeds, they have the starch, and then 6 oxygen is required to make 6 carbon dioxide and 6 water and energy. So that's the one. As as you can see, the amount of oxygen and carbon and hydrogen used for the respiration is exactly available in the production as well. So if you say 1 mole of glucose is used with 6 moles of oxygen gas, and 6 mole of carbon dioxide produced, 6 mole and 6 mole of water is produced. So as you can see, 1 mole of glucose and 6 mole of oxygen, 6 mole of carbon dioxide, 6 mole of water. So if you remove oxygen, right? Carbon dioxide. If you remove carbon dioxide by potassium hydroxide, carbon dioxide dissolved in it, and potassium carbonate can be produced because of that, potassium carbonate. Then let's see, you can see that 2 times 6, that means 2 times 6, 12 oxygen. 2 times 6, 12 oxygen. So the amount of used oxygen will also be removed from the internal air inside the system. So if you remove carbon dioxide, right? Before fixing this one, and now after fixing this one, carbon dioxide will be absorbed in the atmosphere by potassium hydroxide. But when you keep it for few hours, now the seeds are germinating. The germination is started after keeping it in the water for about a few hours. You know that how we put that one, and then take it out and see, keep it there. Wet, nicely soaked seeds in water, nicely soaked in water are used. They are germinating. Then carbon dioxide, that means oxygen, is removed, right? The germination will be continued and will be stopped because the oxygen available in the system will be totally used up, consumed. The oxygen available inside the system will be totally consumed. So no more germination takes place. And as oxygen is used in the system for germination, oxygen is removed from the system. So the volume of oxygen in the atmosphere is 21%. 21% means exactly 20 upon 100. It's exactly like 5 upon 1, 1 upon 5, 1 upon 5. So if you consider the total height is 5, and the water column moves upward for 1 upon 5. And that indicates the oxygen is used for the cellular respiration. Oxygen is used for the cellular respiration. This is visible in this controlled experimental setup, but you can't see it in the control experiment. it is not there. The water level is the same way because oxygen, the germination takes place here as well. But that particular amount of carbon dioxide is available inside the conical flask and the glass tube. So that particular air space is not removed, right? The germination takes place, but cellular respiration takes place because of germination, but because of the carbon dioxide availability, that space is still the same. So by using this one, we can make some questions. We can make some questions. One point that we can use is what are the instruments? What is this instrument? What is this instrument? Right? So this will not be included here. This will not be included here, right? This one is not available. Right. So I can have the questions from here. What is this instrument? What is this instrument? What are these things, right? And what are these things? So why the colored water is used? Why colored water is used? Then what are the observations? Right, you can see, you can write the observations as well. Then, yeah. So there are some questions that we can make, right? Is this clear?
Yes, sir.
Yes, sir.
Anything
Yes,
you
sir.
want to ask before erasing this one? Anything? No? So even this one also can be used in moles reference is there. And this is also can be used. What is the product that might have occur in the boiling tube? Something like that. Okay, this is question. Shall we write the question? I think question number 6 is, it should be question number 6, according to my knowledge. This is question number 4, this is 5. And we are going to write the question number 6. Question number 6. Go, 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 go. Question number 6. Question number 6. Question number 6. Oh. Question number 6. Question number 6. Question number 6. You can see that clearly now. Okay. Cellular respiration. Cellular respiration is one of the characteristics of the living organism. Living organisms given below is a setup is a setup of an experiment. Experiment arranged to test one thing based on the cellular respiration. Cellular respiration. So we want to draw the diagram. We're going to draw the diagram. Conical glass. Then this one. You can have it here. You can have it here. One, two, three, four. And the same one again. You want to draw the same one again. So what I'm going to do is to save my time. I'm going to save my time by having the same one here. Okay. Only the thing is that here you have to change this one and change this one. So I will correct that one and here. Cellular respiration. And yeah. So you can have the part to name this one A, B, C, X, and setup A, setup B, setup A and setup B. Okay. My first question. You can see, right?
Yes, sir.
First question. Part 1. Name A, B, C, A, B, C are parts. Name A, B, C parts. Question number 2. Question number 2. Suggest, S-U-G-G-E-S-T, suggest the suitable living material for B and the things should be done before using in the experiment. Before using in the experiment. Third one. I will put Y here. Y. And then write what is the use of Y. What is the use of Y? A student suggested that the experiment is not correctly set up and one part is missing. One part is missing. What do you think the missing part is? For this is 1, 2, 3. This is fourth one, and this is 5. This is 5. The fifth one. Little slower. 5. Why the water level is risen up to X. Six. Why the water level is risen up to X. How do you come to the expected conclusion? How do you come to the expected conclusion? Mention the conclusion as well. Mention the conclusion as well. 6, 7. Write another observation. Another observation in the experiment. Write another observation in the experiment. Eight. What is the what can be the reasons for your observations? 9th one. Which extra knowledge is required to come to the conclusion of the experiment? And 10th question. 10th question. The last one. In which part of the cell can cellular respiration occur? Cellular respiration occur. Shall I give you more? There are two types of, don't expect the questions like this in your exam. I'm just supporting you some extra knowledge. There are two types of cellular respiration. What are they? What are they? What are they? Next. The colored water is used. Colored water is used in the small container. In the small container. Why or what is the reason for it? What is the reason for it? What is the reason for it? Right, these are the questions that you can answer by using this setup. So there are many questions. Can you answer? I have explained everything. The RAM speed seems to be not enough with my machine. Okay. How much time you want? Finish your answering. Finish. Wow, there are many participants here. Nipun,
Yes, sir.
finish your writing?
No, sir.
Not yet. Okay, let's have a look at the answers. Shall we? Okay. I'm going to discuss the answers. Is that okay? No harm? No response.
Okay,
Can
sir.
I?
Okay.
Yeah. Cellular respiration is one of the characteristics of a living organism. Given below is a setup of an experiment managed to test one thing based on the cellular respiration. Name ABC parts. What are ABC parts? A, A, what is A?
Conical flask.
Conical flask. A conical flask. Conical flask. Then B,
Germinating seeds.
germinating seeds. Germinating seeds. C, KOH solution, potassium hydroxide solution. And the second part, suggest the suitable living material for B, and the thing should be done before using in the experiment. Yep. What is this living thing? Living material for B? It is germinating seeds, correct. But what can be used there? What can you use here?
Germinating green gram.
Green gram, yeah. Or cowpea. Cowpea seeds. Cowpea is not a singular term, it's English one. Cowpea seeds or green gram. And it has part 1, part 2. Suggest the thing can be done before using it. Cowpea seeds, what can be done? Soak in water for few hours. Soak in water for few hours. Then only it starts to germinate, right? Straightway you can't put them. It's just only a few seeds, and the experiment will not be successful. It has to stimulate the germination, right? And so there. Third, what is the use of Y? That is rubber tube. What is the use of Y? Balance the air pressure, balance the internal air pressure when fixing the cork. Balance the air pressure, balance the air pressure of, balance the internal air pressure. Balance the internal air pressure. Balance the internal air pressure when fixing the rubber cork. A student suggested that the experiment is not correctly set up and one part is missing. What do you think the missing part is? What is it? What is the missing part of this one?
Potassium hydroxide in B.
Potassium hydroxide in B. Then both are experimental setups. If you put the potassium hydroxide here, it's the same. I mean, both are the same. The place you touch is correct, but the use of words is wrong. What is the correct word? Yeah.
H2O in experimental setup.
Yeah. The H2O, instead of potassium hydroxide, in C, right, in the boiling tube, here water should be there. So that's the place, right? And right there, the fourth answer, instead of INSTEAD, instead of KOH, same volume of water, same volume of water should be used in set up B. Same volume of water should be used in setup B, that to balance it. The other one, a student, yeah. Why the water level is risen up to X? Why is the water level? Why is the water level? Why is the water level risen up to X?
The
How are you
sheer
going?
types of the oxygen inside the plastic.
Yeah. Little explanation is there, right? Because the O2 is used in respiration. O2 is used in respiration and CO2 is produced. CO2 is produced as KOH remove CO2, the gap of removed O2 is filled by water. Here, used O2 is removed when used here used O2 is removed when CO2 is removed by KOH. Because the O2 is used in respiration and CO2 is produced. As KOH remove CO2, the gap of removed O2 is filled by water. Here used O2 is removed when CO2 is removed by KOH solution. Okay. Can I go to the question? Can I go to the question? Right. How do you come to the expected conclusion? Mention the conclusion as well. Right. How do you come to the conclusion? How do you come to the conclusion? First, you write the conclusion. Conclusion. What is it? Oxygen is used. Oxygen is used for respiration. Oxygen is used for respiration. How do you come to the conclusion? The water level at X is the height of 1/5 of the total height of the total height of glass tube. In atmosphere, 21% oxygen available. So 21% of oxygen is available. So when oxygen is removed, the 1/5 part will be spaced there. So that is filled by water. Moving to the next part. Can I?
Okay, sir.
Write another observation in the experiment. Wow, I expect your answer. Write another observation in the experiment. What is it?
The heat of the flask is increased.
Again?
The heat of the flask is increased.
The heat of the flask is increased. Yeah, I agree. Another one? Another one? That's correct. Another one? I agree with you that answer. The temperature might increase. The temperature of the flask might increase. But what is the other one? In the flask, what can you see? Water?
Paper.
Water vapor. Water vapor. This is, of course, will not be visible or observable, right? The temperature, but water vapor is visible in both flasks. Water vapor is visible in both flasks everywhere. What can be the reason for your observation? If you write the temperature, right? Heat energy is produced by cellular respiration. If you write the second one, H2O is produced. H2O is produced by cellular respiration. Which extra knowledge is required to come to the conclusion of the experiment? Which extra knowledge? These are like for your reference I'm giving. Which extra knowledge? This is 8 and 9. Yeah, which extra knowledge is required to come to the conclusion of the experiment? You have to have some extra knowledge. What is it? What's it?
About heat.
Now the conclusion that oxygen is required for the cellular respiration, but some extra knowledge is required.
Identification of what?
Now,
Amount of oxygen in the environment.
exactly, yes. The amount of oxygen available in the atmosphere is 21%. The amount of oxygen in atmosphere should be is 20% or 21%? 21%. Without that knowledge, you can't come to the conclusion because the height of the water column is risen by 1/5, right? So the total height is divided into 5 equal parts, and one part is risen up by taking the oxygen, removed oxygen. There are two. In which part of the cellular respiration occurs? In which
Inside
organism?
mitochondria.
Mitochondria or mitochondrium. 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 In mitochondrium, there are two types of cellular respiration. What are they?
Aerobic and aerobic respiration.
Aerobic and AEROBIC. Aerobic respiration, and anaerobic, ANAEO, anaerobic, ANOEROBIC, anaerobic respiration. And the next one, the colored water is used in the small container. What is the reason for it? Water and glass tube are, water and glass tubes are, what should I write?
Equally non elaborate.
Colorless, COLOUR, colorless. Water and glass tube are colorless so that water level is not visible unless water is colored. Unless water is not, water and glass tubes are colorless so that water level is not visible unless water is colored or not colored. Not visible unless water is
Not colored.
not colored. Unless water is not colored. What is colored? Unless water is not colored. Okay. See that the time we spent for this part, 1.5 hours period. How are you going to evaluate it, that work? Is that superb or what?
Good.
Yeah.
Super, sir.
Right. Shall I add one part to this question? One part, one more part. That is 1, 2, 3, 13. Write two assumptions. Write two assumptions. ASSUMPTIONS. Write two assumptions used here when conclusion is made. Write two assumptions used here when conclusion is made. Two assumptions. What are they? What are the two assumptions? Yeah. Which one is negligible? The volume of carbon dioxide in atmosphere. The volume of carbon dioxide in atmosphere of flask is negligible. Negligible. We think that carbon dioxide, because the oxygen amount is risen totally because no carbon dioxide is available, we assume. The second one? Second one? What is the second assumption? In respiration, absorbed volume oxygen is equal to released volume of carbon dioxide. It's in the book. The amount of O2 used is equal to the amount of CO2 released. The amount of oxygen used is equal to the amount of CO2 released. Right. That is one part of the cellular respiration. There is another one which is very much common, that experiment to show the released carbon dioxide during the respiration. The experiment is there. Even I can use that one as well. Can I go away from this? Can I?
Okay, sir.
Okay.
Right. The other experiment by using this cellular respiration is that setup. Okay. What is that setup? Now, in this experiment, it is something like this. This is the conical flask where the cellular respiration might occur. And we are going to test that carbon dioxide is released by the cellular respiration. So this is the place where that particular cellular respiration happens. So you can use in this setup, these living materials can be used. You can have an animal such as toad, ගෙම්බෙක්ව පාවිච්චි කරන්න පුළුවන්. Or you can use germinating seed. You can use germinating seeds, right? Or you can use a plant, but it is not that much of successful because if you use plant, the amount of carbon dioxide produced might be taken back by the plant for its the photosynthesis process. If a plant is used, you have to keep the system in a dark room where the light is not available. So you can have the questions like that as well, right? If you arrange the setup and then say, the question might say, one student suggested instead of toad in the particular flask, a green plant is suitable, right? Then write a change that can be done. So then what is the suggestion to have a change there? Keeping the system in the dark room. When it is in the dark room, photosynthesis is not occurring. So it will be only doing the cellular respirations that the plant, so carbon dioxide will be released, right? And now carbon dioxide will be released. So then I'm going to have the taken out carbon dioxide. And then what you have to do is to test whether carbon dioxide is released. Then the only testing method that we can use here is lime water. So you can have the lime water, calcium hydroxide solution, AQ. So the lime water is used. When carbon dioxide is available, lime water turns into milky color. But the carbon dioxide will not, the produced carbon dioxide will not be going smoothly if there is no any pressure. In order to make a pressure, so you have to have another setup like this, which is kept in the water, right? So you can have the water here, and yeah, the water is like you kept, you kept, and water from this one, the water is removed, right? So then you tightly close this one, right? You can pull water from there. You know, hydrostatic pressure can be used. In this one, we are going to study about the hydrostatic pressure in the next lesson. That knowledge also can be used here to prepare the part of the question. The 11th, they know that how water can be taken out, right? Then carbon dioxide, right? So you have to cover this one as well. Otherwise, that air pressure will not be affecting. When water is removed from this tap, there is a gap over here in air. In order to fill up the gap, the air is taken from this space. As the air is taken up this space, the air is absorbed by this one. And now we want to check that the air coming into this one, right, is free. Oxygen should be supplied. Oxygen should be supplied for the germinating seeds. So when oxygen comes, right, when oxygen comes, it should not have carbon dioxide. Okay, oxygen will be given from this system. Oxygen is given by this system, understood? But we want to make sure that the coming air does not have carbon dioxide. So again, you use lime water, calcium hydroxide solution. So if carbon dioxide is coming through this air, this should turn into milky color. And then carbon dioxide should be removed if it is coming. And now we know how carbon dioxide is removed. Which solution should be here to remove the carbon dioxide? The
KOH.
KOH. Right. So now you can see this is potassium hydroxide. This is potassium hydroxide. Okay, KOH is there, potassium hydroxide. The air is taken into the system. Air is taken into the system. The air does not come automatically because of the pressure exerted by water. The water start to move away from this system. As you can see, water is removed. Then there is a gap in here. The air is absorbed by this one, and air comes through this A, B, C, D, E. From the D flask, air is coming there. And to fill the gap, the air is taken from C flask, right? And the C flask has more carbon dioxide now. Carbon dioxide is there because of the germinating seeds or the particular organism, maybe a toad. And then the calcium hydroxide turns into milky color because carbon dioxide is available. But we want to make sure that carbon dioxide is not available from the coming gas here. And we test that whether carbon dioxide is available. This doesn't turn into milky color. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Yeah, you got calcium hydroxide. And potassium hydroxide is available here to remove the carbon dioxide from the coming gases into the flask. Okay, so that is how the setup is set. The experiment is set over there. The question can be asked by using this setup. Is this clear?
Yes, sir.
Anything you want to ask from this part? What sort of questions can be asked? The sort of questions that can be asked, what is this solution? What is this solution? What is the function of it? What is this solution? What is the function of it? And what is this solution and what is the function of it? Suggest another living material instead of given one. what is this solution and what is the use of it, right? And how the pressure or how the system runs, and by using this one, another question can be set, right? Oh, වෙන මොනවද අහන්න තියෙන්නේ? Respiration, irritability and coordination and excretion. Irritability and coordination. Shall I give you one more question? Yeah. From this, that's enough, I believe. I'm going to make the question by excretion. Excretion. The last question for the day. Is that question number 7?
Yes, sir.
Yes, sir.
Question number 7. Question number 7. Excretion. Excretion. Excretion is another living feature of organisms. Number 1. What is meant by excretion? What is meant by excretion? Number 2. Right. 3 excretory product of an animal cell. 3. Which organ of which organ of an animal can remove excretory product. Excretory product. Write the possible excretory product of A. Proteins B. Glucose. Glucose. 5. 5U. Right. Two excretory products of plant. කවුද බොලේ දැන් එන්නේ? දැන් හැරිලා තියෙන්නේ. Write two excretory products of plants and how they are removed. How they are removed. That's enough for the day. And good answers. Okay. අම්මෝ. ෆයිසර් එන්නතේ සාක්ෂරතාවය 86% 143 දක්වා බිඳ වැටී ඇත. Can I? Can I? Yep. Can I discuss the answers? Is fecal matter excretory product?
No.
100%? 100%? 100% no?
No.
No, sir.
Then? Do you think that fecal matter is 100% not excretory product?
Yes.
Correct? Is fecal matter 100% not an excretory product? Your answer is wrong. Fecal matter has some excretory product. What is it? It is the color that gives the yellowish color for the fecal matter that comes from the bile. And bile is produced inside the living cells of the liver, right? So bile is an excretory product. So the fecal matter is not 100% non-excretory product. It is excretory product. It has bile, but not 100%, right? A little amount of bile is available, which is an excretory product. So in fecal matter, there are some excretory products, which is considered as bile. Excretion is another living feature of organism. What is meant by excretion? Removal of?
Excretory products from the body.
You can't use excretory product. You have to define the word excretory as well. Removal of the byproduct of the biochemical reaction occurs in the cell. Within bracket, metabolic activities. What is metabolism? It has anabolism, catabolism. Catabolism and anabolism. Putuwa kadala, atuwa hadano. Putuwa kadala, atuwa hadano. So the breaking of material, the bigger part is we call catabolism. And anabolism is the production of something. So break-in will be producing some unwanted parts, and the chemical process combination will make another excretory product. For example, if you take amino acids at the liver, amino acid NH2 HCOOH. So this is amino acid. Excessive amount of amino acids in the liver will break. It will break this part. Amino part will be broken down, and this one will be used to produce glucose and glycogen. And this one, together with carbon dioxide, produce the urea. It will produce urea, that is NH2 twice. CONH2 twice, that is urea. So the this is catabolism, right? It breaks, the amino acid is broken down into glucose part and amino part. This is catabolism. And this is, from this one, glucose is produced, that is anabolism. And this glycogen is produced, anabolism, and this is catabolism, and that is one way of producing the nitrogenous waste. So anabolism and catabolism. So the answer for that one is, what is excretion? Excretion is the byproduct. According to your definition, I saw that. Yeah. Removal of the material waste or byproducts from the body that are produced during the metabolism. Removal of REMO, removal of byproducts, produced by metabolic activities in the body. Removal of byproducts produced by metabolic activities in the cells is known as excretion. Try 3 excretory products of an animal cell. Carbon dioxide, urea, sweat. Right? Am I
Say that
correct?
H2 done with
H2O you can. Sweat, exhale gas, urine.
salts.
That's how say. Exhale gas, urea, urine, sweat. That also possible. Which organs of an animal can remove excretory product?
Skin,
Skin,
lungs,
Kidney.
lungs,
kidneys.
kidneys. Right. Skin, lungs, kidneys. Write the possible excretory products of protein. I told you, right? What are the possible excretory products? Proteins, urea. Urea, CONH2 twice. CONH2 twice. Deaminification. Amino amlayak hadano. Glucose,
CO2 and H2.
CO2 and H2O. H2O. Write 2 excretory products of plants. Excretory products of plants. 1, oxygen by photosynthesis. And carbon dioxide by respiration. Carbon dioxide and water. Oxygen by photosynthesis, CO2 and water by respiration. And how are they removed? How are they removed? Through stomata. Stomata available in plant leaves. The other one, what's it? Lenticels. Stomata and lenticels. A small small holes, lenticels. Right. How was the day? Fantastic, superb,
Good, sir.
great.
Yes.
And it's worthwhile of coming over here and staying from 5 o'clock up to now. And see you next day. Bye-bye then. We will be meeting.
Goodbye, sir.
Yeah, we will be meeting Wednesday, 9 p.m. to 10:30 p.m. Thursday, 9 to 10:30 p.m.
Thank you, sir. Have a nice day.
Thank you, sir.
Thank you.
Thanks, sir. Goodbye, sir.
Bye-bye.
Thank you.
Take care.